Hello, everyone, and welcome to today's broadcast, Drug Targeting for Inflammatory Breast Cancer, presented by Dr. Kevin Williams, Associate Professor, Biomanufacturing Research Institute and Technology Enterprise, North Carolina Central University. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Williams. I will now turn the presentation over to him.
Thanks, Xavier. So just to give a little bit of background on our institution, North Carolina Central University, we're an HBCU. We're part of the UNC system. We have a strong commitment to health disparities research, and we house two signature institutes. I'm a member of the BRITE, and one of our main missions is to educate students to be ready to work in the pharmaceutical industry. So a quick overview of my seminar. I'm going to talk about targeting IBC through two mechanisms. The first is therapeutic resistance, and the second is the Hedgehog pathway. So a little bit of background on inflammatory breast cancer. And I'm not sure many people have really heard about IBC, but it's a unique, understudied, and very lethal subtype. It accounts for about 6% of all breast cancers, but 15% of all breast cancer deaths. And there's a low survival rate of only 34% versus 87% over 5 years for compared to non-IBC breast cancers. Another challenge is that IBC presents at a late stage 3 or 4 diagnosis. There's no solid tumor, so it's not typically detected by mammogram, hence the no lump required. So no lump required is part of the education campaign by one of our partner groups, the Inflammatory Breast Cancer Research Foundation. So they advocate for IBC. IBC incidence is also higher in younger patients and in women of African descent. The presence of tumor emboli is a hallmark. And again, another challenge is there's a lack of preclinical models and clinical samples. So that's always a challenge. And on the right there, I've got images of a patient with IBC, and the lower right is an image of a tumor emboli from a clinical sample. A little bit more about IBC on the clinical pathology. So clinically, there's a rapid onset of skin changes with progression to stage 3B or 4A within 6 months. So rapid onset is an issue. The breast often looks swollen, red, or inflamed. Or on the right here, it has its pod orange physical appearance. So those are very characteristics of IBC. For the pathology, as I said, one of the hallmarks of IBC is the presence of dermal lymphatic invasion by hyperproliferative clusters of highly invasive tumor emboli. So those are all hallmarks of IBC for diagnosis. Current therapeutic strategies for IBC, multimodal therapy, so neoadjuvant chemo, followed by mastectomy and chest wall radiation. For patients with metastatic stage 4 IBC, if their cancer is hormone receptor positive, they can have hormonal therapy. If their cancer is HER2 positive, they can have targeted therapy with drugs that target HER2. So for example, the tyrosine kinase inhibitor lapatinib that hits HER1 and 2 is one of the few treatment options for IBC, but resistance to that drug is seen in the clinic. So again, another challenge for what we're working on. So alongside Dr. Gayatri Devi from Duke University, which is about 5 miles up the road from our campus, we've been working over probably the past decade on building a program to identify effective therapies for inflammatory breast cancer. So we have a number of grants, we have a number of publications, and I'm going to talk a little bit about a couple of those. Another thing we have a big focus on is disparities in breast cancer. So again, African-American women are typically diagnosed with breast cancer at an early stage, at an early age and a later stage. There are certainly disparities that persist that may involve genetics, biological factors, breast cancer subtypes, poverty, insurance. So those are all factors that we're also looking at. And we're doing that through a couple of training grants that we have with Duke. So I'd like to talk to you a little bit about those. So we have a P20 grant from NCI for a Cancer Disparities Translational Research Partnership. So this is with Duke. So we have a couple of pilot projects, including one on IBC. But I really want to emphasize that we have this cancer research education program that educates our PhD students on biobanking and clinical trial accrual as it relates to underrepresented minorities. And then the second program is we have a grant from Komen on a graduate training in disparities research program. And again, this has a focus on getting our PhD students out of the lab and into the community where they can focus on community-based participatory research. So really helping them connect what they're doing in the lab to what happens with the patient, what happens with the community. So our trainees meet with patient advocates. They learn about human subjects research and IRB protocols. They've been working on designing surveys and organizing focus groups. Excuse me. So one of those was an IBC listening forum that they hosted at the Women's Health Awareness Day on campus back in April. So that's where we had 50 women come in, and we got some sense of whether they'd heard about IBC before, what they knew about it. So that's an ongoing project where we're now going to go out and talk to focus groups and start building an education campaign around inflammatory breast cancer. So to the research I was going to talk about today. So the first one is to look at mechanisms of therapeutic resistance in IBC. So again, this was a collaboration with Dr. Devi at Duke, where their lab has generated a resistance model for IBC. So they did that by taking a breast cancer cell line, inflammatory breast cancer cell line, SUM149, which is one of the few available cell lines that we can use for IBC. And then she did chronic lapatinib exposure. So culture those cells in the presence of lapatinib drug for a few weeks, and you end up selecting out a drug-resistant clonal population, which we term R-SUM149 or resistant SUM149s. So those cells are resistant to lapatinib. They grow in lapatinib and survive. So they've adapted. Then we also went on to develop a cell line where if we remove the drug from the resistant cell line, within about 2 months, we can select a clonal population that regains sensitivity to lapatinib. So RR SUM149 or reverse resistant SUM149. So they regain sensitivity and die in the presence of lapatinib. So that's one of the models I'm going to talk about today. And here's just a snapshot of cell proliferation data. So on the y-axis is cell proliferation percent. On the x-axis is a dose response for the drug lapatinib. So if you look at SUM149s, we get a nice dose response in response to lapatinib. So they're being inhibited by that drug, whereas the resistant SUM149s, they're in purple. Lapatinib has no effect on that cell line. And again, the reverse resistance, we see a nice dose response with lapatinib. So just showing differences in drug sensitivity there. So we were interested in looking at how do these different cell lines respond to other drugs, other tyrosine kinase inhibitors. So what allows us to do that at our institute is that we have a high throughput screening High Content Corp, essentially a small drug discovery company based in their institution. So we have at our disposal over 500,000 small molecules stored on site. So these include FDA-approved drugs, commercial drugs, proprietary drugs. And then we have automation and database systems that allow us to screen large numbers of drugs against large numbers of cell lines. And then we have the faculty and staff expertise from industry. We do medicinal chemistry. We do collaborations, always interested in collaborations. So please email me if you're interested. And we've started a couple of small startup companies. So one of the drug libraries we wanted to screen on these cell lines is the NCI approved oncology drug library. So this consists of 89 drugs at the time that we got from the developmental therapeutics program at NCI. And then we sort of group those by mechanism or type of drug shown there on the left. So we want to screen those 89 drugs in dose response against our parental resistant and reverse resistant SUM149 cells. So here I'm just showing a heat map readout for SUM149 and resistant SUM149 cells. So again, this is cell proliferation data. We're dosing the drug over a 10-point dose range. Green means there's 0% inhibition. Red means there's 100%. And I know it's a little difficult to read there, but I got lapatinib highlighted there, where we see that it's sensitive to the SUM149s, but has no effect really on the resistant. So what was interesting about this profiling of these drugs was that we saw a group of drugs, which are shown there clustered at the top by that bracket, that sort of had cross-resistance. So there was a set of drugs that were also resistant in the resistant SUM149. So that was a group of targeted chemotherapeutic drugs. So it wasn't just lapatinib alone that showed resistance in that cell line. So that was sort of interesting to us. So if you plot IC50 values for the parental SUM149s and the resistant SUM149s, so these are some of the drugs that show dramatic differences between those two cell lines. We see big increases in IC50, so inhibitory concentration value of 50% inhibition for drugs that include chemotherapeutic agents and also tyrosine kinase inhibitors. So maybe about a dozen total where we see this sort of cross-resistant phenotype. So if we then go on and also add in our reverse resistant SUM149 cell lines, again, if you look at the top there around the bracket, we see that those cells regain or resensitize to all of those targeted and chemotherapeutic drugs that were resistant in the R SUM149s. So we go from sensitive to resistant to sensitive for that collection of compounds. And again, if you look at IC50 values between those three isogenic cell lines, you see again the IC50 value goes up for the resistant, but then comes back down for the reverse resistant as they regain sensitivity to each of those drugs. So again, this was sort of unexpected and interesting to us. As I said, there are chemotherapeutic agents there are tyrosine kinase inhibitors. So they have very diverse mechanisms of action. But if you read a bit more about them, they are all reported to induce reactive oxygen species, or ROS. So we started delving a bit more into that area. So oxidative stress and redox adaptation in cancer. So oxidative stress is an imbalance between ROS and the cell's ability to remove ROS, or its antioxidant defenses. So chronic exposure to increased ROS stimulates protective mechanisms or a redox adaptation. So that promotes therapeutic resistance. So essentially, oxidative stress acts as a selective pressure to promote the survival of cells that can detoxify the increased ROS. So we looked at some of these mechanisms in our cell lines. So one thing we wanted to measure was ROS production. So on the left there, I'm showing an assay that we use to measure ROS. So percent cells with ROS staining is on the y-axis. So in the white bars there are the parental SUM149. So you can see that if you put lopatinib on those cells, which is shown with a plus mark there, you see a big increase in ROS. However, if you put lopatinib on the resistant SUM149s, that's the black bar, you do not see that increase in ROS. So somehow they've adapted. If you look at the reverse resistant SUM149s in the gray bar, after a couple of months, they have also regained sensitivity to lopatinib, and you see a big increase in ROS when you treat those cells with lopatinib. So part of that is to also look at expression of antioxidant proteins, so catalase and superoxide dismutase. So we see that both of those proteins are increased in the resistant SUM149 and decreased back down in the reverse resistant to levels seen with the parental cells. So that's some Western blot data. We also did some more Western blot data to look at some apoptotic pathways. So we see activation of pro-survival stress response signaling in the resistant SUM149. So if you look at the Western blot there, we see that XIAP and IAPs and survivin all increase in the resistant SUM149 compared to the parental. And then those levels are decreased back down in the reverse resistance. So those proteins are all part of the inhibitor of apoptosis pathway. So what do we think is going on? So we think that our SUM149 cells are cross-resistant to those drugs due to their suppression of the oxidative stress response. So those resistant SUM149s have adapted, they have redox adaptation, they've increased antioxidant capacity, they've increased survival factors, so those cells can survive in the presence of the drug. They do not die. Whereas the reverse resistant cells, they decreased survival factors, they decreased antioxidants. So when we add drug to those cells, we see big increases in ROS and subsequent cell death. So, we're interested on where those cells come from, where do the resistant cells come from. So we and others have proposed that there's a subpopulation of reversibly drug-tolerant cells that are dormant until they see drug or see high concentrations of drug. So there's a subset that tolerate growing in the presence of the drug indefinitely, and we call these drug-tolerant persisters. So if you look at the schematic in the upper right there, on the left is essentially the parental SUM149 cells that have both sensitive cells and drug-tolerant persistent cells. And if you expose that cell heterogeneity to a TKI like lopatinib, we see only the drug-tolerant cells emerge and survive. So the suggestion is that these drug-tolerant clones exist de novo and may share some properties with cancer stem cells. And SUM149 cells do show significant ALDH positivity, a marker for cancer stem cells. And we see even higher increase in ALDH in the resistant SUM149 cells. So we're investigating a lot of these mechanisms here, the role of cancer stem cells, drug-tolerant persisters, and that's an ongoing project. But I'm happy to share more about that if you have questions. So to get to the second part of the seminar, so this is targeting the Hedgehog pathway in inflammatory breast cancer. So the Hedgehog pathway has a central role in the growth of a vast array of cancers. So on the left here, it's showing all the cancers that have a role, the Hedgehog has a role in that cancer. And on the right is a schematic of the Hedgehog signaling. So the Hedgehog ligand binds to the patched receptor and relieves inhibition of the smoothened receptor. Then GLI transcription factors are activated and translocate into the nucleus and turn on various transcriptional programs. So there are drugs that are on the market that target smoothened and inhibit Hedgehog signaling. So those are essentially out there for basal cell carcinoma. But again, acquired resistance is seen in patients taking these smoothing-directed drugs, so another challenge. So my lab is interested in GLI1 activation in breast cancer. So GLI1 is one of those downstream effectors of Hedgehog signaling, and there's a lot of evidence out there now that GLI1 has a critical role in the progression and invasiveness of multiple cancers. So GLI1 promotes an aggressive breast cancer phenotype. Nuclear expression of GLI1 is a strong predictor of poor prognosis in many cancers, including breast cancer. And GLI1 expression correlates with poor survival in breast cancer patients, especially with the basal subtype. So on the right there is some survival data from patients who either have low GLI1 expression or high GLI1 expression, and those are patients with basal-like breast cancer. So we've looked at GLI1 expression in many cell lines. Here I'm just showing a set of breast cancer cell lines where we've done qRT-PCR to look at messenger RNA expression of GLI1. And what we see across this panel of cell lines is that GLI1 is highly expressed in basal-like IBC and basal-like non-IBC breast cancer cell lines. We've also shown previously that if we knock down GLI1 in SUM149 cells, there's a reduction in cell proliferation. So how is GLI1 activated? It's activated by both canonical and non-canonical mechanisms. So canonical is through the Hedgehog smoothing pathway that I just showed you in that earlier slide. There's also quite a lot of literature now that other pathways can activate GLI1 through what's termed non-canonical mechanisms. And indeed, there's a few papers out there now showing that GLI1 activation in breast cancer cell lines is predominantly Hedgehog smoothing independent, so non-canonical. And we've shown that for IBC, where those cells are not Hedgehog responsive. So in most breast cancer cell lines, it seems like there's a non-canonical activation of GLI1. We published some of that back in 2011. So for a number of reasons, we're looking at targeting at the level of GLI1. So downstream of smoothing, there's a lot of interest in developing GLI inhibitors that would both inhibit canonical and non-canonical mechanisms. So we're interested in looking at GLI antagonists that block GLI activation. And there are a number of GLI antagonists that have been identified from GLI luciferase reporter screens. So the HPIs shown here block GLI activation. They can block GLI1 translocation to the nucleus. JK184 acts in the nucleus to block GLI-mediated transcription. The GANTs, like GANT58 and GANT61, act in the nucleus to block GLI1 as well. Excuse me. So we did a study where we wanted to profile a set of these GLI antagonists in multiple models of IBC. So this is just really a snapshot of quite a lot of data from one of my Ph.D. students. So in the upper left panel there, we assayed all these inhibitors in dose response in a Hedgehog functional cell-based assay. And we saw that they all inhibited Hedgehog signaling to varying degrees. We then took a subset of those compounds and looked at their inhibition of proliferation in a panel of breast cancer cell lines. We looked at how those drugs could inhibit colony formation of breast cancer cell lines, and 3 of those were reasonably potent in that model. We looked at a couple of those drugs, GAN61 and JK184, that had dramatic effects on GLI transcriptional targets and cell cycle progression genes. So, we were trying to sort of triage out what were the most effective or efficacious GLI antagonists that we might want to run in animal models. So we also looked at GLI antagonists in a high throughput 3D spheroid model. So this is where the cells are growing as spheroids that we think is more representative of a tumor and more predictive of in vivo efficacy. So we have a format where we take 384 well plates that are ultra low attachment, and we can seed cells in each well and we get a single spheroid that forms in each of those wells. We can then use high content imaging to look at various effects of drugs on spheroid formation. So the upper panel there is just a shot of one of the 384 well plates, or part of it, where we've done a dose response of two of those gli antagonists, JK184 and GAN61. We've got a dose response. We're running them in replicates of 5. And we're looking at effects on spheroid area, spheroid fragmentation. Just below that is zooming in on those spheroids after they've been stained with Herxed and Yo-Yo-1 stain. Herxed stains for DNA. Yo-Yo-1 stains cells if they are dead. It can only get into a cell if it's dead. So we can image those spheroids at different drug concentrations and look at different effects of different drugs. And as I said, some drugs shrink the spheroids, some drugs fragment the spheroids. So we're using various analytical methods to analyze those effects. So we can quantitate some of those effects. For example, at the bottom there, I'm showing a couple of graphs. So one is looking at spheroid area after we dose on GAN61, and we can generate an IC50 for that drug in terms of how it affects spheroid area. On the right panel there, effects on spheroid Yo-Yo-1. So we see an increase above a certain concentration where we see that the cells are dying. So we can generate a lot of data on a lot of compounds in this model. And we've also developed, along with Dr. Devi, a tumor emboli, high throughput tumor emboli model. So I mentioned when I was talking about IBC that the presence of tumor emboli are a hallmark of IBC. So we've developed a model where we can grow some 149 cells as essentially tumor emboli. And then we can put drugs on those models and see effects. So for example, if we treat those tumor emboli with GAN61, we see a dramatic decrease in the size of the tumor emboli. So that's shown in the images there on the right. So all this data really told us that GAN61 in particular was a very interesting drug that was a gli antagonist and had dramatic effects in our panel of phenotypic assays for IBC. So we took that drug in vivo in a mouse model. So we had a mouse where we put some 149 cells in the memory pad of the mouse. So it's an orthotopic model. The tumor grows, and then we start treating with either vehicle or GAN61, dosing every few days and measuring the size of the tumor every few days. So on the upper left panel there, I'm showing some 149 tumor progression in that mouse model, where on the y-axis is tumor volume as it's increasing as the tumor is growing, and on the x-axis is the number of weeks of treatment. So you can see that if we dose the mice with GAN61, we get a significant decrease in the size of the tumors, and that's shown in the picture below. We also see dramatic effects on GLI1 protein expression and GLI1 messenger RNA expression. So again, GAN61 was having a dramatic effect in this in vivo model. So it looks like it's a promising compound. So what also is interesting about GAN61 is it's been shown to bind directly to GLI1 protein. So there's a few publications down there in the bottom right. And so here I'm showing a schematic of GLI1. It's a large protein. It's 1,106 amino acids, over 100,000 molecular weight in size. So the zinc finger or ZF domains are shown there in brown on the schematic. So like I said, it's recently been shown that GAN61 binds directly to the zinc finger domain, in particular zinc finger 1 and zinc finger 2. So, we're interested in this mechanism. We're interested in seeing if targeting the zinc fingers of GLI1 is a viable strategy for moving forward. So our lab is interested in GLI1. We're looking at multiple strategies for developing small molecules that will target GLI1. And it is a challenge to find drugs that target transcription factors. That's why it's unusual that GAN61 binds directly to GLI1. Transcription factors typically do not have what we call druggable sites. They don't have nice pockets where we can get a small molecule in there that binds tightly. So you can take sort of alternative approaches. So we can look at blocking DNA binding, sort of what we're doing with GAN61. We can also look at blocking nuclear translocation of GLI1. And we can also look at inhibiting activating kinases. So there are a number of kinases that phosphorylate GLI1 to activate it. So again, we could target those kinases and hence target GLI1 activation. So one of those kinases that we're interested in is DYRK1A, or dual specificity tyrosine regulated kinase 1A. So there's been a number of publications out there showing that this kinase promotes nuclear localization of GLI1 and stimulates GLI1-dependent transcriptional activity. So on the left panel there is some data from one of the publications below, where they've taken NIH 323 cells, excuse me, and they transfect in a GLI1 GFP reporter, and GLI1 remains in the cytoplasm. If they then co-transfect in DYRK1A kinase, that GLI1 then moves to the nucleus. If you take a DYRK1A construct that's kinase dead, so it has no activity, GLI1 stays in the cytoplasm. So that was pretty strong evidence that DYRK1A is promoting GLI1 nuclear translocation. So we were interested in that for sure. So one thing we were interested in was whereabouts on GLI1, as I said, that's a very large protein, does DYRK1A phosphorylate GLI1? So one of those publications had shown that GLI1 was phosphorylated, but it was in two reasonably large fragments. They didn't really narrow down whereabouts that phosphorylation event was. So we were interested in identifying that phosphorylation site. We were going to do it using phosphopeptide mapping and mass spectrometry. And that's a collaboration we did with Dr. Lee Graves at UNC Chapel Hill. And this was work done by Ben A., who's a student in my lab. So we essentially did this using an in vitro kinase assay. So we incubate GLI1 protein with DYRK1A protein in the absence or presence of ATP, in the absence or presence of a DYRK1A inhibitor called harmine. So we incubate those, we then run them on an STS-page gel. We cut out the bands that are GLI1, so that's shown in the black boxes there. And we do a tryptic digest of the protein in that band. We enrich for phosphopeptides and we do mass spec. So on the right is showing a mass spectrum of, in the absence of ATP and in the presence, and we see a shift in molecular weight that corresponds to a phosphorylation event. And when we look at our samples, we see that phosphorylation events only occur in sample 3. That's highlighted there with a red circle. So that's the sample where we have GLI1 plus DYRK1A plus ATP. So that's not unexpected. But what we did see, we only saw one sequence that was phosphorylated. So that was interesting. So serine 408 was the only phosphorylation site we saw that was phosphorylated by DYRK1A. So if we look at that phosphoserine 408, it falls within the putative nuclear localization sequence of GLI1. So that sort of makes some sense that DYRK1A is phosphorylated in a site within the NLS, and hence it has a role of impact on the function of GLI1 and how it might translocate into the nucleus. So we have obviously ongoing studies looking at mutating that. So if we mutate that phosphoserine site to another amino acid, does that then block GLI1 translocation? That would be our prediction. So, our lab has a big interest in drug discovery for the Hedgehog pathway. So we're interested in seeing if we could target DYRK1A with small molecules to block GLI1 activity. So Can we find an inhibitor, maybe have a very selective chemical probe to block the pathway and potentially turn it into a drug long term? So, our hypothesis is we have an inhibitor of DYRK1A that blocks GLI1 translocation into the nucleus and hence blocks GLI1 activation. So we've developed a high throughput screening assay for this kinase. So this consists of purified enzymes, and we're using what's termed TR-FRET. So the DYRK1A kinase has a GST tag, glutathione transferase tag, and we have an antibody to the GST that is europium linked. We then have a tracer that binds in the ATP binding pocket of the kinase. So that's shown on the schematic there on the right. So in that condition, the tracer and the europium are in close proximity, and you get high FRET, you get a strong signal. If you inhibit the kinase, or you have a drug that blocks tracer binding to the kinase, you don't have that close proximity of the tracer and the europium, so there's low FRET, so that's inhibition. So that's essentially how that assay works. That's where the window is. So we've run a number of screens on DYRK1A. I'm just showing a little bit of that data. So one of the high throughput screens we did was using a FDA approved drug collection of 1,280 compounds. That's termed Presswick. So we screen each of those compounds at a single 10 micromolar concentration. So each well has one drug, and it's just a 10 micromolar. So on the right there at the bottom is a typical high throughput screening scatter plot that we use to look at the data. So on the y-axis is inhibition of the kinase activity. On the x-axis is the number of wells. So each well is a different compound. So in the red and blue, we have our controls. The red are our 0% inhibition controls. The blue are our 100% inhibition controls. We have those controls on every plate, obviously. And then the green circles are each compound that we've screened. So if we take a cutoff of 50% inhibition, so anything above 50% we term an inhibitor, we see that there are 23 compounds with greater than 50% inhibition. So we've confirmed those by dose response, and we're looking at running those in a number of assays to see if they've got potential as inhibitors, Hedgehog pathway inhibitors, GLI activation inhibitors. So just to finish up, we've taken a number of strategies to target GLI1. We've shown that DYRK1A phosphorylates GLI1 within the nuclear localization sequence. We've identified some inhibitors that target DYRK1A. We're also looking at GANT61 still and its role in blocking zinc finger DNA binding, and that, again, as a potential drug. So really summary from the Hedgehog part of my talk, we've shown that GLI1 is highly expressed in basal-like breast cancers, including the IBC. GANT61, a direct binder of GLI1, has activity in multiple IBC models and activity in vivo. So we're currently running studies that target novel mechanisms of GLI1 activation. And as I said, we're testing our DYRK1A inhibitors in a number of cellular and in vivo models, hoping to publish some of that data soon. So I'm just going to finish up, acknowledge a number of people in my lab, Mike, Maria, Helen, Shalanda, and collaborators, Dr. Debbie and Dr. Graves, Dave Dar at UNC, and also funding from DOD, the NCI, and the Susan G. Komen Foundation. And as I said, we work very closely with patient advocates from a number of IBC foundations. So Ginny Mason at IBC Research Foundation. So I'm going to finish there. Thank you, and any questions?
Thank you, Dr. Williams, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the send button. Our speaker will follow up with your questions via email. Now let's get started. Our first question is, what strategy could you take to eliminate the emergence of drug-tolerant cells?
Yeah, so that's a good question. So I think we showed that in those drug-tolerant cells or our resistant SUM149s that there's increases in survival pathways. There's increases in those anti-apoptotic pathways. So, we're interested in maybe looking at drugs that block those pathways. So we think that if we treat the parental cells with lapatinib in combination with drugs that target those redox adaptation pathways, we can eliminate the emergence of those drug-tolerant clones. So we need to do some sort of combination there where we knock out, the sensitive cells and also knock out those tolerant cells at the same time. Thanks.
And is GANT61 a viable drug candidate?
Again, that's a good question. There's a very recent paper showing that the active form of GANT61 is actually a fragment of the structure I showed in that earlier slide. So we're still looking at it from a medicinal chemistry point of view. There's not been a lot of data done preclinically on GANT61. So, the typical sort of toxicology study, I haven't seen anything published on selectivity of that compound. So I think that's still an open question. Thanks.
And for our last question, are you concerned about selectivity of your DYRK1A inhibitors?
Again, that's a good question. There are kinase inhibitors that have been FDA approved that have varying selectivity. Some of them hit multiple kinases. Some are reasonably selective. We think we might need a reasonably selective DYRK1A inhibitor. There are some very closely related kinases. There's DYRK1B, 2, 3, and 4. There are CLKs, which are another kinase family, very closely related. So we're trying to understand how much selectivity we need. Do we need to have something that's an absolutely selective DYRK1A inhibitor, or if it's a sort of pan DYRK inhibitor, is that sufficient? So that's part of our ongoing studies. Thanks.
Well, I'd like to once again thank Dr. Williams for his presentation, and I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January of 2019. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.